Hi there, my name is Ibanda, a member of the Systems Medicine Group based at the Department of Pharmacology and Therapeutics of the University of Liverpool in the UK. Today, I'll discuss two features that are critical in setting the right target compound profile for a successful lead optimization process. To, to do this, I'll focus on how we define an ideal compound profile, develop strategies to achieve it, and apply modeling approaches to illustrate how we can accelerate drug discovery and development. The target compound profile, or TCP, defines the characteristics required for a molecule to progress successfully into drug development. It has key parameters, including potency of biological activity measured by metrics such as IC50 and EC50, selectivity, which looks at the off-target activity, toxicity, which could also look at the cytotoxicity, pharmacokinetics, physicochemical properties such as the partition coefficient, and the pharmaceutical properties for developability assessment. Defining a TCP early establishes clear progression criteria and ensures that the discovery activities remain aligned with the desired clinical outcome. Therefore, the TCP defines the drug-like properties that are required to achieve the desired therapeutic outcome, and this guides the compound design, selection, and optimization. However, selecting the right biological target remains the most critical step, and therefore confirming that the target is genuinely relevant to the disease is essential. As such, this slide illustrates how target validation and evaluation can be performed. The example focuses on CETP inhibitors which failed in multiple clinical trials despite successfully modulating lipid biomarkers in phase 3. In hindsight, the researchers could have strengthened their target validation process by first confirming adequate CETP occupancy by the inhibitors such as tocetrapib, and then evaluating whether this target engagement translated into meaningful biomarker changes. Validation is achieved when target binding leads to the expected biomarker response and desired efficacy. Conversely, if a chemically distinct compound achieves adequate target occupancy and biomarker modulation but fails to demonstrate efficacy, this provides evidence against the target and could have invalidated CETP as a therapeutic strategy. Therefore, our goal is not simply to modulate a target or alter a biomarker. Rather, we seek compelling evidence that links the target directly to the disease process. A robust understanding of the target's biological role helps define the minimum pharmacological properties that candidate compounds must achieve within the TCP. This slide introduces the background for the modeling study presented here. Numerous studies have shown that exposure of candidate-based oxidizing agents such as hydrogen peroxide disrupts the cellular redox balance, resulting in oxidative stress and subsequent DNA, lipid, and protein damage. Depending on the severity of the stress, cells may undergo cell cycle arrest at either G1S or G2M phase, or ultimately die through apoptosis or necrosis. To investigate the mechanisms governing cell fate in candle beacons, we developed a conceptual model based on two competing hypotheses. First, we hypothesized that both the G1S and the G2M checkpoints remain active under oxidative stress. Second, we proposed that the oxidative stress induces cell death independent of cell cycle phase. By quantifying the transition rates and the death rates, we aim to determine how oxidative stress influences cell fate decisions. This slide shows experimental data collected after 4 hours of exposure to varying concentrations of an oxidizing agent, and this is as reported by Wan and colleagues. As we can see, as the concentration increased, two clear trends emerged. First, the proportion of the dead cells increased, and there was a progressive accumulation of cells in the G2M phase of the cell cycle. These observations suggest that cell cycle arrest and cell death may be regulated independently rather than sequentially. We subsequently tested 4 alternative mechanistic models against this data. Each model was a distinct representation of a particular cell fate mechanism by Kandal beacons. The best performing model supported that the G2M checkpoint is critical and suggested that apoptosis affects only the G1 and the S phases of the cell cycle. This graph summarizes the concentration response relationships predicted by the best performing model. We can see that as at lower concentrations, the cell cycle arrest dominates, becoming apparent at about 0.6 micrograms per milliliter, and then dropping to 0 very sharply. As the concentration increases, cell death becomes progressively more prominent, eventually reaching a maximum of about 14 per hour units. It is important to note that the predicted concentration ranges associated with the cell cycle arrest, which is an adaptive response, and cell death are clearly separated. This suggests that these two are distinct biological mechanisms. And to summarize this modeling part, we can say that although this model does not incorporate direct measures of target engagement, it demonstrates the value of mechanistic and mathematical modeling for understanding complex cellular responses, like what we've seen in terms of response to oxidative stress. But overall, we can see that combining experimental data with mechanistic modeling can save time. It can also reduce uncertainty and improve the decision making throughout the drug discovery process. In conclusion, we can say that TCP is the set of drug-like properties needed for a successful therapeutic candidate. Target validation will help us generate a target compound profile by providing the biological evidence that is needed to define the properties that a successful drug candidate must possess early in the discovery process. And lastly, modeling is an important time-saving strategy during TCP generation.